Good evening, everyone, and thank you very much for joining us on this next edition of the SIP of Science series, supported by Semmelweis University Translational Medicine, Center for Translational Medicine, and the Department of Anesthesia and Intensive Care. And today we have a precious guest, Professor Peter Biro. He's originally from Transylvania, so he could speak Hungarian, but he won't. He will speak to us in English, which is the official language of the SIP of Science series, because we have guests from outside Hungary as well. So Peter is a professor in anesthesiology, and we know each other for quite a long time now. He has had several research fields. And today he picked this topic on neuromuscular transmission blockade and monitoring, which is very important, as we all learned over the last few years, and this seriously affects outcome after surgery. So with this introduction, Peter, it's a great pleasure to have you here, and I hand over the stage to you. We already see your slides, we can hear you, so please proceed. Thank you very much.
Thank you very much, Zsolt. Köszönöm szépen a kedves meghívást. But I will stay with the English language because I don't know the Hungarian technical terms in this topic. So I think this is, in any case, a little bit better. But I'm ready to answer questions which are posed in Hungarian. So, as far as I understood, most of the people listening to me are intensivists, but probably a good part of them are anesthesiologists, and I speak from the anesthesiological perspective. But whatever I have to say, it has also some relevance in intensive care. So let's see what is going to happen. So my conflicts of interest, there are quite a lot of them, but only one is relevant in this context. I'm medical advisor for RGB in Madrid, who is manufacturing a neuromuscular transmission monitor, which I will also mention here, so please be aware of being biased. But otherwise, I speak more of experience and general knowledge. We will hear some details about the difference between qualitative and quantitative aspects of neuromuscular transmission monitoring. By the way, neuromuscular transmission monitoring or relaxometry, the older term, are interchangeable. I will speak about prevailing problems with this kind of monitoring up to date nowadays, what technologies are now on the market, general characteristics of these techniques which are available. Then I will speak about an ideal monitoring, how I would like to have it in its best optimal form, and some goodies from my clinical experience. So let's start with this topic, qualitative versus quantitative aspects of neuromuscular transmission monitoring. So what is the difference? Qualitative, on the left side, are this tactile or visual watching of the movement of fingers, and that is not that exact. For example, you cannot distinguish by these qualitative measurements a train of 4 of 0.6 from 0.8 or 0.7 from 0.9, and this is absolutely important and decisive. On the other hand, when you use a numerical scale of the depth of the blockade, then we speak of a quantitative neuromuscular transmission monitoring, although these numbers are on ordinal scale, not really mathematically calculable, but they are of a big help. Now, many anesthesiologists don't even use at all neuromuscular monitoring, transmission monitoring, and they usually say, I never used it in my career of 40 years and I never had problems. These people are wrong. They had problems, but they didn't realize them. So still 20% of anesthesiologists in Europe, 10% in the United States, and even more in other countries never use neuromuscular monitoring at all, not even qualitative. Those who use, nearly half of them use still the visual and tactile method, which is really insufficient. It is like driving the car with this kind of vision, as you see on the right side of the slide. So then those who finally who use the quantitative measurement, a big part of them use only trainer 4 as a stimulation pattern and don't know that trainer 4 only covers superficial and moderate block. Sometimes they even don't know about what is below, and there's quite a lot. There is the tetanic count region, which goes from 0 up to infinite, but it makes sense to measure it up to 15, 16. From then on, it starts to overlap with the trainer 4 count. So I hope you are all familiar with these three measurement patterns. Trainer 4 ratio, the most superficial one, is measuring the index between the first and the fourth stimulation, the reaction of it. When the fourth disappeared, you have still 3 counts, then 2 counts, then 1 count, and then 0 counts. And when you have a 0 count of trainer 4, you are still not in a deep block. The deep block is measured with post-tetanic count, which is a stimulation, tetanic stimulation of a few seconds, then a little break, and then come one after the other, 15 or even more single twitches. And the number of twitches which elicit a movement or electromyographic signal in the muscle, these are counted as positive numbers. Only when even PTC doesn't elicit absolutely no reaction, then we speak of a total blockade, which is actually never useful and should be avoided. So what I propose is, let's see the whole spectrum now of, to make it elegant, I propose 30 steps, which are beginning with trainer 4 ratio of 100% or 1.0. Then we have here 10 steps of trainer 4 ratio, 0.9, 0.8, 0.7, and so on. Then when the fourth is disappearing, the fourth reaction, you have trainer 4 count, 4, 3, 2, and 1, and 0 trainer 4 count is already overlapping with a higher number of post-tetanic count, approximately between 12 to 15. It depends on the individual. And then you have, let's say here, 16 steps of post-tetanic count, which gives a beautiful total of 30 steps. And these 30 steps I prefer to use as a spectrum. Now, on the right hand, you have no blockade at all when you didn't give blocking agent, and the other end is the total blockade where you have not even a PTC reaction. In general, one can say the blocks are like this: superficial blockade in the upper TOF ratio region, moderate blockade is very broad from the lower trainer 4 ratio to the upper or mid PTC region. Deep block is PTC of 4 to 8 or 10, and profound block is 1 to 3 PTC. You will see this is a very important region which sometimes is necessary to achieve. Now, for anesthesiologists, all these deep blocks are not important, and probably this is the reason why anesthesiologists don't know about PTC in many cases. They need, for safe intubation, a trainer 4 count of 2 or below. A trainer 4 count of 2 is enough. And for a safe extubation, according to the guidelines, you need a TOF ratio of 0.9, but this is already disputed. It still has the ability to produce complications. Actually, we need a TOF ratio of 1 to extubate safely the patient. So anesthesiologists, they like to move in this region. But when you give an intubation dose, usually the blockade goes far beyond the TOF count of 2. It goes somewhere into the PTC region, even maybe to PTC 0. But if you do not top up it, then the patient will come out of it and moves slowly back to the right side. Now, for the surgeon, actually superficial blocks are irrelevant. He doesn't need them. Either he doesn't need a block, or if he needs a block, he needs at least a moderate block, which is somewhere in the low TOF ratio region into the upper PTC region. This is very good for general anesthesia in traumatology, for example, to make the joints easily movable, but that's still no complete blockade. The diaphragm can still contract and would do so if the patient is provoked to cough by a not so deep anesthesia. And if you want to be really sure that the patient will not move at all, complete or adequate immobilization, you have to go to this ominous PTC of 1 to 3, but avoid 0 and avoid going above 3, which is really difficult. You will see that it is not easy to maintain blockade in this region if the surgery requires it. And many surgeries don't need at all blocking, but some are profiting from the intubation dose, which is still there. The surgeon starts the operation, let's say, within 20 minutes, and then the patient is still partially blocked and will not move. So the surgeon will be even happy then because there is still some residual blockade. But otherwise, for the anesthesiologist, residual blockade is a nightmare. He doesn't want it. So to safely extubate, I mentioned you need a TOF ratio of 1. Now let's look how useful are these blocking regions. To have the lower TOF ratio is completely useless, not good for anesthesia and not necessarily good for surgery. The surgeon wants it here for a good moderate block. Then we have another inadequate blockade. If you want to have a totally immobilized patient, you cannot leave him in the PTC 4 and above. That's simply not enough. So that's inadequate deep block. And what you need is this profound block between 1 and 3 PTC, and avoid 0 block. And of course, when the patient is going to be waken up, avoid residual blocks in the range of 0.7, 0.8, 0.9, even 0.95 TOF ratio. Insist on TOF ratio of 1. So what happens if you just give boluses to maintain the blockade? You will have repeatedly movements to the left on the scale, as you see here, with these triangles. And as soon as you don't give a drug, the patient goes back to the right because he is metabolizing the blocking agent. So it's always constant motion, except you make an infusion of the blocking drug, which is not yet very widespread. I learned to use it in neurosurgery when we had a long period of blockade, deep blockade necessary, and I can tell you it works very nicely, but you must measure. You cannot do it without monitoring. Now I spoke about this very deep, profound blockade of 1 PTC to 3 PTC, why it might be necessary. There are some surgeries where it is very good to do it in order to avoid even movements by the diaphragm, which is very resistant to blocking agents. Sometimes you even cannot block it completely at all. So here is just an example of several studies which have been made in a long period of the last 10 years. This one has compared a moderate block of trainer 4, 0 to PTC above 4 versus a profound blockade for PTC between 1 and 3. And what did they find? They have in the profound block less intraoperative movements. Probably they didn't give such a deep anesthesia. And probably it was an IV anesthesia and not an inhalational anesthesia. And the patient have less postoperative pain. And why is this? Because they don't need such a high pressure in the insufflation of laparoscopic operations. So whenever you do laparoscopy and you insufflate CO2 into the abdomen, tend to use as little pressure as possible, because the higher the pressure, the more pain the patient will suffer postoperatively. And when you use a deep block, a profound block, you need less pressure to fill up the cavity. So that is the rationale behind this argument. Now, the problem with the PORC, which is actually postoperative residual curarization or postoperative residual blockade, that should be avoided, of course. One, that the patient can breathe well, but it's not about hypoventilation. Above 0.4 train of 4 ratio, there is not a big problem to ventilate, to breathe, of course, for normal patients. Some patients with pulmonary disease I exclude from this argument, but it is something else. We will come to this back to this problem, a problem which may cause pneumonia a few days later, after the patient has returned to the ward or into the intensive care unit. And this is totally out of the view of the anesthesiologists. They don't see the patient on the 4th day. And that's why they have long time ignored this problem. And for many years, a TOF ratio of 0.7 was declared sufficient for emergence of anesthesia. It has been, meanwhile, recognized that it is wrong. And 0.9 is now the actual threshold in the guidelines, but it is already disputed severely. And I personally, I'm on the opinion that that's not enough. It should be 1.4. Now you see here on the left side, typical operations when you really need a very deep, profound block in ophthalmology, in airway surgery, laparoscopic procedures, in particular the robotic surgery and some neurosurgical operations where little motions of the patient, for example, during a cuffing, can destroy an eye or make a damage in the brain. So there is a need of adequate immobilization in some cases. And in the other cases, you want to avoid postoperative residual curarization. And you see very nicely on this image here on the right side that with a train of 4 ratio of 0.4, the forced vital capacity is not anymore that much reduced. For a healthy individual, it doesn't matter. And the tidal volume is nearly normal. So there are many other clinical signs which come back, but this one is the real problem, the airway obstruction. One might think, if the patient has a blocking, a residual block, how can he obstruct the airway? He cannot close his vocal cords. That's true. The vocal cords are very sensitive to the neuromuscular blocking agent, but many other hypopharyngeal muscles are extremely resistant. And because he cannot close and might aspirate a little bit of saliva, he starts with a laryngeal spasm, with the hypopharyngeal muscles. So in this situation, when you have a little bit of residual block, you have the high risk of airway obstruction. This sounds not plausible, but it is proven. I have seen it many times when the patient was extubated too early. So quantitative measurement is absolutely important to tailor the blockade according to surgical needs, particularly when the surgeon wants a deep block, and to enable safe emergence from anesthesia on the other end. So, and what happens in between is not so important, but it is important to maintain these two situations whenever they are needed. Now, neuromuscular transmission monitoring is not so easy. There are a lot of problems. For example, one of the biggest problems is the huge inter-individual response, pharmacokinetic response to a given drug. Please imagine all these little black triangles are individuals who have received an intubation dose of rocuronium only and nothing else. And then there were many TOF ratios recorded, from each individual, and you see a huge discrepancy. Some return full transmission in 20 seconds. Maybe they got a little bit of smaller dose, but they are very fast metabolizers. For example, people who had a burn. And others are extremely slow, some which have probably some enzyme deficiencies. In any case, you never know from your patient to which category he belongs. He can be a fast metabolizer, moderate metabolizer, or a slow metabolizer. He is, for example, one patient who after several hours didn't return above 0.6 TOF ratio. I mean, this happens. This can happen. So this is a strong argument in favor of measuring. Another problem, in contrast to pulse oximetry or capnography, which are global parameters, they collect the data from the whole body. Neuromuscular transmission monitoring, relaxometry, is strictly local. It's only measuring on a very limited neuromuscular complex. So that's really a problem. And because the neuromuscular complexes are different, sensitive to neuromuscular blockers, you have to know that whatever you measure is maybe representative, but not exactly the same what you want to know. And there are only two organs, let's say, or not only, but mostly two organs are important, is the glottis and the diaphragm, which are totally different. You can see here in the recovery profile after rocuronium that there is 15 minutes difference between different muscles. Diaphragm is very fast. Interestingly, the laryngeal muscles, except of the vocal cords, is fast too. And then abdominal wall and others are here. And you see the AP, the adductor pollicis, where it's measured since decades with the classic measurements. They are late. But it's true what you measure. It's simply the measurement of a neuromuscular complex at a specific place. And that's it. And it doesn't say that other muscles are at the same status. So whatever you measure at the wrist, usually the other muscles have already recovered. And this is the problem with the airway obstruction, which I meant. On the one hand, residual block still can make airway obstruction, but the patient cannot close with the vocal cords, and then he undergoes a risk of aspiration. And the follow-up of risk of aspiration is the pneumonia, which usually begins 4 days later. So what we are interested in are the glottis and the diaphragm, and they have completely different recovery profiles. With acceleromyography, you are in the late phase. With the compressomyography, you are somewhere in between, and that's it. Now let's talk about these different technologies. There are many more than the mentioned two. We have mechanomyography, which is really measuring the port, but this is not available in the clinical routine. It is more experimental and lab, but it is the gold standard. Whatever you want to measure in comparison, you compare it with mechanomyography. And the wrist, but again, what I mentioned, it has limitations concerning the other muscles. Then you have acceleromyography. This is probably one of the most widespread ones, has dominated the market for relaxometric devices for decades. It has been a little bit partially replaced by kinemyography, which is measuring also the direction in which the thumb is moving, but it's still a wrist-based measurement with stimulation at the ulnar nerve and the reaction of the adductor pollicis sensing it. Now, electromyography is rather new. It's only since about 8 years on the market. It doesn't measure any more movements. So it is immune to positions of the hand that doesn't matter anymore, but it measures the electromyographic response of the adductor pollicis muscle. And the problem is it is just a little bit above the background noise of electric stimulations. And because of this, it is prone to artifacts and disturbances, for example, by the coagulation during the surgery. Then we have phonomyography. That is the only technique which would be in a multi-local measurement, not really global, but multi-local. They put microphones on different parts of the body, but this is not really feasible in everyday routine. And what is the newest and latest is the compressomyography, which is measuring the reaction of muscles in the upper arm under a cuff, which is also used for blood pressure measurement. And it stimulates these muscles directly by electrodes in the cuff. I will come back to this method. So these three techniques are really those who are usual accelerometry, the most widespread one, the newer electromyography, and the compressomyography, which is represented by only one device. Here in this slide, which I don't want to read all the details, but I have collected the disadvantages in red and advantages of these three techniques. And as you can see, they are a little bit different, and it matters also for the local preferences, what you would like. So I personally prefer the compressomyography, but as I mentioned, I am biased because I have collaborated. I still collaborate with the company producing it, but you will see I know a lot of it about it, and that's why I'm talking about it. So they have different displays, different modes of action, but let's say the most complete one, the compressomyography, will be mentioned here in the following slides. So my views about an ideal monitor are not yet accomplished, but I believe that this device here comes closest, but as mentioned, I have a conflict of interest, but as a co-developer and extensive user, I have a lot of experience with it and I can talk about it. Also about its disadvantages. So it works like this. You use a regular blood pressure cuff, which has here 2 electrodes inbuilt. You have to put them on the upper arm, but you can put them also on the leg. You have 2 upper arms and 2 lower legs to disposal, and you just have to check that the electrodes are in the medial side of the upper arm. And then you place it and you can measure blood pressure with it, but it can also do neuromuscular transmission measurements, which are lasting less than 30 seconds. For each measurement, it is inflating the cuff a little bit to 50 millimeters mercury, and then it stimulates, and it detects the pressure variation in the cuff because of the muscular movement. It has, you use it usually in a so-called autopilot mode, which means you only start it and you must not touch it anymore. It does everything by itself. So you give the hypnotic to the patient, he falls asleep. Then you start the device in autopilot mode, which will make an in vivo calibration. And then you give, after the in vivo calibration, so 20 seconds later, your intubation dose of relaxant. You do handbag ventilation until relaxation is sufficient, like in a 4 count of 2, which would be here somewhere. And then you can do the intubation. This autopilot feature is very useful because you must not touch the device anymore until the end of the surgery and anesthesia. It always knows which kind of pattern it should use, whether trainer 4 or PTC, and when it can do it, it knows that it is not permitted to do too often PTC because that produces a post-tetanic potentiation. And as soon as patient is recovering, it will increase the frequency of measurements and will show you at the end and you are on the safe side. So I just skip this. You see the whole trend with very easily interpretable icons, ratio decreasing, the 4 TOF counts are available, then only 3, 1, 0 TOF count. From this time on, the device starts measuring PTC. This is a 0 PTC after an intubation dose. And then you see the recovery. You give a bolus. You can place here a little sign. It goes back to 0 PTC, but then soon after it comes out. And when the surgeon is unhappy with the recovery, then you give again a bit of a procuronium and the patient returns to a lower level, but then he will recover completely towards the end of surgery. And now I come to a few goodies from my clinical experience. One case which is remarkable is a Da Vinci prostatectomy, where I really like to use this technique. You see here an excerpt of the paper protocol. Duration of anesthesia was long enough. I used a total of 200 milligram of rocuronium, 50 milligram for intubation, and 150 for repetitions, because I wanted to keep the patient very low at a PTC of 1 to 3. You will see is not so easy. So the time-related dosing was 33 milligram per hour. Now, when do you need, in such a case, the block? You need it during pneumoperitoneum only. And you see, after induction, the TOF ratio diminished, TOF count went away to 0, and we had some positive PTCs, but the patient recovered quite fast. And when the surgeon started with the insufflation, then the patient got a higher dose, 10 and afterwards 5 milligrams of rocuronium, and he went back down. And now I was busy to keep him in this green box, which is set by myself. I can set to the range. It's better visible where I am. And you see, with repeated bolus doses, it was not so easy to keep him under PTC of 3. Actually, this upper limit is PTC of 6. I was very generous at that time. Now I'm not anymore. And you see, the 0 PTC, which is unnecessary, happened in 27% of the total pneumoperitoneum time, which is not very elegant. Only 50% were optimal, PTC of 1 to 3, with boluses. Some were acceptable, 12%, below 6, and too light were 11% with a PTC above 5 or above 6. So you see, it's not easy to keep the patient with repeated boluses, which are quite often given, but you can do this when you measure continuously, and the autopilot always knows what to measure when, so you don't forget. I can say 50% was optimal, 50 not so optimal, but 89% was safe. The patient will not move with the diaphragm. So this can be probably improved by giving the drug with an infusion pump and adjusted according monitoring. TCI is not necessary, but you can do even closed loop or semi-closed loop control with the measurements. So this was my presentation. I have also to mention there are some problems even with the compressor myography as it is nowadays, which I have to mention. It competes with non-invasive blood pressure measurements. Sometimes you need to wait a little bit. Then it can slow down the infusion flow if you have the measurement on the same arm and the IV line peripherally, because of course with these repeated measurements there is a pressure on the cuff and that slows down the venous return. It needs still better published validation, which has been done already but not yet published, and it needs to overcome the dominance, the tradition and dominance of wrist techniques, because it's simply more representative. And it needs to be compatible with various PDMS systems, patient data management systems. There are some already, as far as I know, with Philips, but more is to be expected. And now I finish the lecture. Thank you very much. Please open your microphone. I don't hear what you say.
Can you hear me now?
Yes.
Oh, sorry, I pressed the button, but there was a bit of a delay. So, a great lecture. Thank you very much. And excuse me. And trust me, all of the audience are anesthesiologists. So,
okay.
All of us are anesthesiologists. So, your points, I'm sure they were well taken. And we just had a different webinar on neuromonitoring. And the importance of intracranial pressure monitoring was discussed most of the time, which is again something that we believe that we know, but we don't. And I think, and we just discussed it during your presentation with Laci Zubek, who is my deputy and sits here in the studio with me, and attention to details. I think that is a key issue nowadays in current anesthesiology. Do you agree, Peter?
Absolutely.
It's not about bringing the patient out alive from the operating room, but attention to details. And it's unbelievable how important, how serious damages can be done to our patients by not monitoring things. And I encourage the audience, there are quite a few here, to post questions in the chat, and then we can answer those. But neuromuscular monitoring was the main topic of your talk. But what do you think about entropy, so depth of anesthesia monitoring? How complementary is this with neuromuscular monitoring?
If it would be reliable, I would like it to have. I used actually bispectral index for a long time, and not entropy, but they are doing the same. I think it is important, but for another reason. This post-anesthesia problems in older patients, they can be minimized if you do not bring the anesthesia too deep. But if you give a lighter anesthesia, then you risk awareness and you risk movements if you do not do blocking of the neuromuscular transmission. So if you want to make an elegant anesthesia, you use entropy or bispectral index. You keep the patient at a level of 50, for example, 40 to 50, and don't go below, but then you have to block the patient in order to avoid movements. When you block the patient, you will never see if he has an awareness. So that's why it is important.
Exactly.
Yeah.
This will be my second. If you do a post-tetanic count of 1 to 3, then I think measuring depth of anesthesia is really important.
Absolutely, because you can't keep him with boluses, as I have shown you. I tried many times. The patient very often evaded my range and had to be reblocked and reblocked again and again. So I emphasize to use continuous infusion in those patients where you need a constant immobilization.
I have only a short question. Which is the ideal administration method, continuously or with the boluses? What is
for usual normal surgery, boluses are absolutely sufficient when you don't need to keep the patient in that narrow band stripe of 1 to 3 post-tetanic counts. In moderate blockade, it doesn't matter if it is a post-tetanic count of 7 or 10 or TOF count of 3. It's still good enough for the surgeon. For example, if he's doing some limb surgery and wants to move freely the arms or the legs, that's enough. But when you are down there in the cellar, then you need an infusion to keep the patient up there. And you have to adjust the infusion. And don't forget, the reaction of the system is slow. So you must make big changes in the infusion rate to see an effect.
Is neuromuscular monitoring compulsory in Switzerland?
Yes. Meanwhile, it is mandatory to use it. But it's not said which kind of measurement you use, and there are still many using qualitative measurements or outdated wrist measurements. But you can improve. You always can improve.
I was very much intrigued by your slide indicating that the same intubation dose of a neuromuscular agent had a completely different effect.
Yes, it's incredible. There is no other drug of which I know such a range. Not opioids, not propofol, nothing. In opioids, you can have a double dose. Or in patients who are addicted, that's something else. But it's in the range of twofold, maybe threefold, but not 20-fold as it is with neuromuscular blocking agents. But fortunately, these very slow metabolizers are not so often. So most people are in the range of where you can expect that the written half-time is correct. For rocuronium is 40 minutes, for example, or 45 minutes. In the most cases, it is around that, but not in all.
Now, do you have data on more robust outcomes regarding inadequate neuromuscular monitorings? What I mean that we do have data that if we don't measure blood pressure continuously and it is hypotension occurs, the more it occurs, the longer it lasts. It has serious post-operative myocardial, cardiac dysfunction effects. If you don't measure depth of anesthesia, it has post-operative cognitive or delirium effect. So do we have similar clinical outcomes, outcome data on not using neuromuscular agents appropriately?
There are such papers. I can't name now one, but you must consider that there are. I have to check, and I can send you that later. But what is important to know, the really serious problem because of residual block is pneumonia, which starts a few days after anesthesia. And this happens still rarely. So maybe in 7, 8, 9, or 10 cases per 1,000 anesthesias. So it's a rare occurrence, but it's very severe. And very rare occurrences, which are severe, are difficult to quantify. You need huge numbers to find a significant difference. For example, in pneumonia, if the patient has had monitoring or not. But I'm sure such papers exist, and I will be able to deliver.
Yes, well, fortunately, anesthesia has become extremely safe. This is why, regarding hard outcomes, we need tens of thousands of patients to show any difference in any case of intervention.
That's true.
Yeah.
Well, but there is, of course, a special patient population which is very vulnerable. The older ones, the frail ones, those with a reduced lung capacity, and we have more and more patients of this kind because the aging is going on, and more old people are still operated. And we should not forget this problem and not only concentrate on cognitive problems, which are also a topic, an important topic, but also simply the safety of the airway.
Well, Peter, I really enjoyed it, the discussion as well. Thank you very much. And to the audience, the lecture will be available in the next tomorrow. Attila, our great help, just reassured us that tomorrow it will be on YouTube. So you are more than welcome to see and distribute it, and we will also distribute the link within our mentoring community. There are more than 1,000 participants. So, Peter, thanks again, taking your time. It was great to see you and listening to you. And we will see the audience of SIP of Science in 2 weeks. Thank you very much. Bye-bye.
Thank you. Bye-bye.
Bye-bye.